Good afternoon, ladies and gentlemen, and welcome to the 13th ÖGMBT annual meeting from molecular machines to translational medicine. This year's organizers are our scientific community members in Innsbruck in Tyrol. My name is Lukas Huber, and I am the president of the Austrian Society of Molecular Biotechnology, and I welcome you wholeheartedly to our online meeting. Now, we are the largest life science organization in Austria. As such, we organize and support our scientific community. We have therefore the responsibility and the mandate in difficult times to keep up scientific community structures, the dialogue and the discussion about science, about our results. Do they care about our young members and about our community? Our meeting is now the second time in a full online format. And this is quite remarkable because last year we were the first scientific organization in Austria which went online during the crisis in an online-only format with such a broad dimension. We enjoyed very much doing it, and that made the difference. We didn't complain, but we enjoyed doing it because we discovered there are many advantages associated with an online format. And we did it to our best knowledge and abilities. Of course, we made mistakes, but we learned from these mistakes that we did last year. And from that, I can assure you that this year, we serve our community again with even better tools and better concepts. Professor Farhan from our Innsbruck scientific community will tell you in a minute what the structure and the organization of this year's event will be all about. So why should you be a member of this scientific community? Now, it has many advantages to be a member of the ÖGMBT. You get information, updates, a newsletter, information about grants, about career events. You get access to a magazine, the Chemie Report, who is our official magazine, where we have always a full article in the interest of the community and the interest of our society, where we support the FWF, where we support scientific structures, where we bring in front of the curtains scientists from our community. We have a meeting calendar for our members, and we support the job market, and we have a further education board. This links me immediately to ILSA, which is the heart of our society, the young life scientists in Austria. They are extremely active. They organize all career events. They organize excursions to pharmaceutical companies, to research institutes, all kind of events to meet each other, to come together. This generates a network from memberships that we have where we reach out to Europe, like FEPS, FEMS, and others, connections to our stakeholders, and the membership network itself. We engage and participate in the public dialogue about science. We communicate the needs of the community to the politicians and to the funding organizations. And the central part of our meeting is always our annual meeting, where we have this interdisciplinary exchange. Now, again, it's free for all members to participate in that online format. We supply also travel grants, and we have awards. We give awards to the best short talk, to the best poster. And what we also have is we have the scientific awards this afternoon, the ceremony, where also the Ministry of Science will participate with representatives. And this brings me to the acknowledgements and to thanking all the people involved in the organization of our annual meeting. At the forefront is our scientific committee headed by Professor Hasso Fahan in Innsbruck with all his board members. And they are all not only excellent scientists, they are excellent community caretakers, and they organized a wonderful event with extraordinary speakers. I have to, of course, thank our sponsors, the ministry, that you see here on the top left corner. They support us generously with money for these very important awards. We have a silver sponsor again since many years from the pioneers of biotechnology, Professor Kattinger with the polyimmune. And we have a number of companies that you see listed here that very generously supported us. We have a media partner that supports us. And of course, this is the Chemie report. Now, with this, I have to thank also our back office, because the back office is the backbone of our organization. And as today, they are managing everything in our daily life, our communication within the society, our reach out to other societies, to the public, and they are organizing from the technical point of view and from the organizational point of view, this meeting in the background. And I would like to thank all of them very much. You are a marvelous team. And with these words, I will stop my screen sharing and hand over now to the meeting organizers in Innsbruck. Please, for your words.
Thank you very much, Lucas, for your words and for also giving me actually the chance to organize this. It's now almost a year ago that when I was moving from Norway to Austria, that Lucas said, this is like your welcome present. You're going to organize the next annual meeting. Like this, you get to know the local community better. And I was really looking forward to have a meeting in place in Innsbruck with a lot of people, because I heard normally this meeting has between 300 to 500 people in Innsbruck. So I was really looking forward to it. However, the pandemic situation did not relax as fast as we were all hoping. And we had to take this very difficult decision with the president and with all other members of the ÖGMBT, and made a decision, with heartbroken, but we had to go for an online meeting again. All of us were looking forward to have a meeting in presence here, but we had to make this decision. And I'm happy now that we did it now in retrospect. Nevertheless, we were a bit afraid that this will, the participation will drop down. Nevertheless, we have managed to attract 32 plenary speakers. We have 83 short talks. And what do I have here? 58 flash presentations. So this makes already a very, very nice bunch of people that are going to share their scientific results with all of us. I was really, I didn't organize the meeting myself alone. I had an excellent community behind me, that session chairs that were responsible for their sessions. They really did it completely on their own. They didn't bother me at all with anything. So it's really very nice to have such chairs that support you when organizing this meeting. I hope that this meeting will be a huge success. Despite the fact that it's an online-only meeting, it's not a hybrid. We decided not to go for a hybrid solution. Nevertheless, I hope that this meeting will nevertheless allow you to participate and interact with each other as much as possible. We also offer this year, for the first time, presentations for the industry. We have 6 industry presentations to give also our generous sponsors the ability to present their scientific results to all of us. With this, I am closing. I wanted to have only a very, very brief speech myself. And with this, I'm handing over to Professor Fleischhacker, the rector of our medical university.
Well, thank you very much, and good afternoon from my side as well. I would also like to extend a very warm welcome from Innsbruck and welcome you all to the annual meeting of the ÖGMBT. By the way, in the practice session, Hasso Fahan had mentioned weird acronyms. This is probably one of the biggest challenges of my term here as president of our university, to memorize acronyms of various different institutions, also institutions at our own university. Just as a little by note, the CCB, as it's well known, that the host of this meeting today, is an acronym that not everybody really can translate into real words. And even more difficult is the name of the department that is hosted in the CCB. And an informal poll from my side a few months ago, I found that only very few of the active professors knew in which department they were actually working. I do realize they know the name of their collaborators and various other people there. So acronyms are a tough story and give a lot of raise for concerns, but also some jokes here. I would like to extend from my side a warm thank you also to the local organizing and of course also the international organizing community, and especially the hard work that the scientific committee has done to make this meeting possible. As you know, the Medical University of Innsbruck has very tough safety rules regarding COVID-19. And I would just like to mention here that in this respect, at least, is a perfect meeting, that most of you are probably vaccinated or recovered from the disease. Plus, you're all online, so the risk of contagion is basically reduced to zero, which perfectly fulfills all our safety rules that we have here. Nevertheless, I must say that it's a real shame, despite Luca's enthusiasm for online and web-based meetings, that we cannot host you in person here in beautiful Innsbruck. And we do hope that this will turn out differently. So better luck next year. Finally, I wish you all a fruitful academic exchange, also following the Society's goal to fascinate young research. So enjoy, listen carefully, learn from each other, enjoy the interaction, and maybe come up with new brilliant ideas, new interesting stories about the science that fascinates all of us. Good luck with the meeting and have fun.
Thank you. Thank you very much, dear President of our university. It's an honor and great pleasure for us that you join in today and share this welcome with us. We are very thankful for that, and we are very much looking forward now to a very exciting meeting. So, according to the program, you are now guiding through in a flash talk way for the different sessions, and I hand over to you again.
Okay, I will be just calling up the sessions by number. So we start, and when I call you, you just start sharing your screen, or if you don't have a presentation, you just don't do that. Session number 1: chromatin and gene regulation.
Yeah. Macht, aber ich sehe jetzt nicht das. Einen Moment. Okay. Okay, so welcome everyone from our side. First of all, I would like to thank Hesso as the organizer, and also the speakers that all join us, are going to join us in the next few days. And most of all, maybe the people behind all of this, the people at the ÖGMBT in Vienna who made all this happen, actually. So I think we have a very nice lineup of speakers to kick off this meeting tomorrow with the first session, chromatin and gene regulation. So we will have 2 invited speaker sessions plus a keynote speaker and several short talks. So we will start tomorrow with a talk from Andreas Adorna, who is the chair of the new institute formed by the LMU in Munich, And he and his group, they are interested in chromatin and gene regulation, and in particular in chromatin remodelers. So he will talk tomorrow about the structural study, basically, how one of these chromatin remodelers called ALC1 is regulated. So ALC1, like all chromatin remodelers, is regulated by cofactors or co-regulatory domains. And this ALC1, as you can see in this small slide, is recruited to sites of DNA damage. And Andreas Adorno will tell us tomorrow how this interaction of ALC1 with its cofactor, which is this NAD metabolite, how this interaction is important for regulation of ALC1. So the second talk that we will have tomorrow is given by Dr. Matthias Munschauer, who is recently, or more or less recently, appointed junior group leader at the Helmholtz Institute for RNA-based infection research. And as you all know, viruses always exploit the cellular machinery for their replication, and this is in particular true for RNA viruses, which use or make use of the host cell RNA binding proteins. And Matthias will tell us tomorrow about a recent study they did, where they tried to map the interaction of cellular RBPs with the SARS-CoV-2 RNA in infected cells. And they have generated a kind of map, and they also use several techniques to show which of these interactions are then crucial for the infection. And as you can imagine, this may even have therapeutic potential in the future. And on top of these two invited speakers, we have several very interesting short talks, and I will just give you here this overview slide to tell you what you will hear tomorrow. So we have a slide about nuclear architecture and neurons. We have a talk about the regulation of chromatin states and their impact on the developmental potential of oocytes. We have a talk about gene regulation by a long non-coding RNA fungi. And we have two talks about epigenetic regulation in cancer, one dealing with the DNA demethylation enzyme and one with the function of a histone deacetylase. Then we have two talks that deal with two different types of histone variants. One is SAMP-A, which plays a role in mitosis and chromosome segregation, and one is histone H3.3, and we will learn here about the role of this histone variant in the brain. And last but not least, we have a talk about DNA double-strand breaks in meiosis and how these DNA double-strand breaks are formed and how they then implement how recombination events in meiosis can take place. And now I hand over to Alexandra Lusser.
Yes, hello. Also from my side, I'm Alexandra Lusser, and I have the pleasure to announce to you the first of 3 keynote speakers of this meeting. And this first speaker, we are very happy that we can welcome Professor Bas van Stensel. And we have a slide here. I'm sorry, it seems not to work to move on. Yes, here it is. So this is Bas van Stensel. He's a professor at the Netherlands Cancer Institute. And since my group also works in chromatin regulation, I have known his work, of course, for many years. And I believe many of you might as well, because Bas is one of those rather rare, I should say, scientists who contribute to the progress in the field, not only by many important discoveries. In his case, it was about nuclear architecture, higher order chromatin. But his group has also been very busy over the years in developing multiple really crucial techniques that had a tremendous impact on the whole field and brought us forward. And I believe that one of those methods, the most prominent one, that is called DEM-ID, some of you might have even used yourselves to map protein interactions with the genome. So as you can see from the title, Bas is going to talk about the very fascinating question of how specific locations within the nucleus, like the nucleolus or the lamina, have an effect on gene regulation. And I'm sure it's going to be a very exciting talk. And I hope I will see you tomorrow at 6 p.m. when his talk starts. Thank you. And back to you, Hazel.
Thank you very much. So then we move on to the next session that is headed by Markus Keller and Katrin Watschinger. It's Cell Biology and Biochemistry of Lipids.
Yeah, thank you. Hi, my name is Katrin Watschinger. Before getting into detail about the session that my colleague Markus Keller and I are chairing tomorrow afternoon, I would like to thank the Austrian Association of Molecular Life Science and Biotechnology, obviously especially the back office. This is Andrea, Walter and Zrinka, as well as Hasso Farhan, our local organizer of this year's meeting, and all our colleagues from the organizing committee for their help and enthusiasm in compiling such a thrilling meeting to which we are all now looking forward to.
Yeah, also from my side, hello. Katrin and I are the chairs for session 2. We both share a strong common interest in lipids, and this will also be the topic of this session. Katrin is interested in the biochemistry and physiology of isolipid. I by myself, I'm looking into regulation of membrane lipid metabolism in rare inherited metabolic disease. And in line with this research interest, we were really excited about the opportunity to organize this session for you at this ÖGMBT meeting. Of course, we would have loved to have it in person, but we are also happy with this virtual form. Our session will take place tomorrow, starting at 1:15 p.m.
So our session, as Markus already mentioned, focuses on lipids, many aspects of lipids. This ranges from basic research in lipids, state-of-the-art lipid analytics, and goes then to metabolism in health and disease. And these topics will also be covered by our two excellent plenary speakers, which are Ruth Birner-Grünberger. She's a professor for chemical technology and analytics at the University of Technology in Vienna. And Ruth, she will speak about lipid hydrolysis in cancer. And our second plenary speaker is Stephen Claypool, who is professor for physiology and genetic medicine at Johns Hopkins University in Baltimore. And Stephen will talk about mitochondrial membrane musing.
What we quickly want to show you here on this grid for our lipid session tomorrow afternoon is that beside of these two excellent plenary speakers, we have put together what we think nicely balanced and diverse program, covering a lot of the topics that are of interest from a lipid perspective, but also from many other levels of analysis. This program also perfectly shows that lipid research in Austria is very well positioned and highly active, as we have flash talk and short talk presentations coming from 7 different universities, from Graz, from Vienna, and from Innsbruck. We are very much looking forward to meet you at our lipid session tomorrow, and we are looking forward to have stimulating discussions and highly interesting presentations. Thanks a lot.
Bye, see you tomorrow.
Thanks a lot. So I will call now the chairs of the third session, which is again Alexandra, Sebastian, and Matthias. The session is on RNA biology.
Yep, so.
Michael?
Well, did I turn? Yep, my micro is on. No.
Yes, we can hear you.
Ah, perfect. So welcome from our side, and it's my pleasure to invite all of you on behalf of all speakers and organizers to session number 3 about RNA biology, which will be on Wednesday, starting at 1:15. And this session will cover a wide range of different topics. So I didn't start. Something is. It covers a wide range of topics on RNA biology. So it's organized in 2 plenary talks, 6 short talks, and also 3 science flashes. And the session will start with the first plenary speaker, which is Claudia Höbartner from the University of Würzburg. And her talk is entitled Ribozymes Meet RNA Modifications. And she will tell us about the beauty and functionality of nucleic acids, as she will show some work on RNA enzymes that can modify RNAs, and also DNA enzymes that can detect RNA modifications. So this will be a really exciting talk on this topic. And the second plenary speaker will be at 4:15, and it will be given by Stepanka Vaniachova from the Central European Institute of Technology in Czech Republic. And she will talk about the spatial intactome of 2 very important RNA modifications, which is 6-methyladenosine and 6-demethyladenosine, and how they try to disentangle interaction network from the modifications and they involve proteins by close proximity labeling and quantitative mass spectrometry. So this will be a really exciting talk from that side. But besides these 2 exciting talks, we also have 6 short talks and 3 science flashes on a different variety of topics on RNA biology, covering piRNA processing, microRNAs and microRNA processing, RNA labeling, decoding, splicing, RNA protein interactions, and RNA preparations. So I hope to see you on Wednesday and to witness a really exciting RNA afternoon with all these talks and contributions. So with this, I'm already done and hand back to Hasso. Thank you.
Thanks a lot, Matthias. So now we move on to the next session, which is on biophysics and structural aspects of signaling proteins. The chairs are Theresia and Martin.
Hello, everybody. I'm very happy that we can announce now our session 4, where we will include different techniques to understand much more on a molecular and atomic detail how signaling in general works. And I hope you can see now our blue screen with the ÖGMBT logo. Our session will...
No, we can't see your screen.
Sorry. Does it work now?
Yes.
Okay, sorry. So... sorry, start again. So our session will start on Wednesday afternoon on 1:15, and we will have 2 plenary talks, and I will introduce Thomas Leonhardt, who is our first plenary speaker. Thomas Leonhardt has a very strong background in analyzing signaling processes involving lipids, so he will explain basic principles how signaling works, and on the left you see those electric signs for closing and opening circuits, how transistors work and everything, and he will translate that into cellular events, especially function of lipids in signaling. His lab is at Max Barrett's, or is part of the Max Barrett's labs in Vienna, and he will be online tomorrow as our first plenary speaker.
And our second plenary speaker tomorrow will be Patrick Ayers from the University of Liverpool in the United Kingdom. He will introduce us into the function of human pseudokinases, which are kinases, as we know, which lack their expected biological function due to mutations. And he will have a specific focus in his presentation on the so-called dark kinome, which covers pseudokinases for which the biological functions are currently very mysterious and still unknown. And he will present his latest results in this field. He will also talk about how pseudokinases regulate and affect signaling mechanisms, and how these molecules, how these proteins can be targeted by small molecules, which might act as a future drugs in the near future. In addition, we have a couple of short talks. We have 3 short talks and 6 science flashes, which cover a variety of different topics, ranging from enzymes to membrane proteins, antibodies, signaling in prokaryotes. In terms of techniques, many of these or some of these presentations are focused on computational chemistry, but we also have a lot of experimental results which will be presented tomorrow in these together 9 presentations. So we are very happy to have such an exciting program on Wednesday, and we're looking forward to seeing all of you in our session Wednesday afternoon. Thank you.
Thanks a lot. So next in line is actually myself. It will be the session on proteostasis. Just start the presentation. So in the session on proteostasis, we will have 2 speakers, one junior group leader, actually, someone, Elif Karagöz, who started her lab only 2 years ago at the Max Perutz laboratories in Vienna. And she will talk about phase separation and the unfolded protein response, which I think is a very new and very trendy topic in this field. The second plenary speaker will be Eric Chevet from Rennes in France. And he is actually, his laboratory combines the investigation of basic cell biology like ER stress and cancer, in particular on glioblastoma multiforme, which is like one of the cancers that he's mainly investigating. In addition, we have 6 short talk speakers that cover all the range of the academic ladder that you can think of. So the first speaker, who is a very young PhD student from the Medical University in Innsbruck, will talk about a new post-ER quality control mechanism. Then we have someone that will talk about Golgi stress that comes from the University of Essen in Germany. Then we have a postdoc from my laboratory, although it's written here Oslo. I moved myself recently, so I still have part of my team in Norway. So Zahur will be talking about the role of ER stress and splicing, the connection of RNA splicing and ER stress. Then we have a PhD student just about to finish who will talk about the role of lipids in autophagy. And then in the end, we have 2 really young group leaders who are even younger, like academically, than Elif. Elif started a lab 2 years ago. Aid and Kai started the laboratories just a year ago, and we are giving them the chance also to present their past results, but also the future plans in this area of research. And finally, we have also 5 flash presentations, all from Austrian universities. So overall, we are combining here 5 different countries in this little session that we will have on the 23rd of September. Thank you very much. And I hand now over to Zrinka, because the pitch for session number 6, Immunology, is, I think, pre-recorded, right, Zrinka?
Yes, it's pre-recorded. Just a second.
Welcome, everybody. My name is Verena Labi, and I'm running the Experimental Immunology Group at the Medical University of Innsbruck. My lab is interested in how B lymphocytes generate cell fate and how cell identity is disturbed in diseases like autoimmunity and cancer.
And my name is Natascha Kleiter. My group focuses on how nuclear receptors regulate protective versus destructive effector and memory function in T cells in different disease settings, such as infection, cancer, or autoimmunity. I'm happy to introduce the first plenary speaker of the immunology session, who is Matteo Iannacone from the Dynamic of Immune Response Unit at the San Raffaele Scientific Institute in Milan, Italy. He will give us insight on his fascinating work regarding the functional biology of hepatic CD8 T cells. As although the liver is the target of several infectious, inflammatory, and neoplastic diseases, thereby affecting millions of people worldwide, analysis of in-depth T-cell responses and their dysregulation in disease has only recently started. So join us in this exciting topic. We have also invited 3 fantastic young scientists to present short talks. First, we will have Leslie Calderon from the IMP in Vienna, who will tell us how PI3 kinase signaling is regulated in B cells to ensure antibody-mediated immunity. Next, Michaela Luciano from the University of Salzburg will discuss how the NLRP3 inflammasome drives myeloid leukemias. And finally, Franziska Werner from the Medical University in Vienna will provide insights into the role of memory B cells in melanoma growth.
And we will also have interesting flash talks from 5 early career scientists from all over Austria, actually, to cover a wide range of immunological topics, for example, autoimmunity, thrombosis, or allergy. And our second plenary speaker is Susanna Minguet from the University of Freiburg. And Susanna's group studies how signaling events are initiated at the plasma membranes of lymphocytes. And her basic research also informs the design of chimeric antigen receptors on T cells, so-called CAR T cells, which is a promising strategy in cancer therapy. And we are looking very much forward to Susanna's presentation, which we believe is a really timely topic. And of course, we are looking forward to meet all of you for the immunology session Thursday afternoon this week at 1:15. So see you there.
Thank you very much. So we move on to session number 7, cell proliferation and cell division. Stefan and Marin.
Okay, can you see my screen?
Yes, if you, it's kind of, yes, now we see everything.
Okay, I would like to welcome you and invite you to the first session in the coming week on Tuesday the 28th at 1:15, to the session of cell proliferation and cell division. We will have 3 main speakers who will cover aspects of mechanical aspects of mitosis, as you could briefly see in the movie. I don't know whether the movie is still running. Doesn't seem to be the case. Here you see the beauty of mitosis, and the beauty of mitosis, the smooth machine of the mitotic spindle that controls chromosome segregation. The essential players are well known, but how these proteins, microtubules, microtubule-based motoproteins, kinases and phosphatases cooperate to make this process work and guarantee the equal segregation of chromosomes is by far from completely understood. So we'll have 3 main talks covering the mechanic aspects of the function and regulation of the mitotic spindle, as well as 1 talk concerning the spindle assembly checkpoint. So this is the response of cells to a perturbed mitotic spindle. And if you're interested in pseudokinases, as I mentioned in session number 4, there will be another topic covered in this presentation. In addition to our 3 main presentations, we'll have 8 short presentations who either cover aspects connected to these 3 main topics, But in addition, they will also discuss cellular consequences of failed mitosis. So either if things go wrong in mitosis, that might lead to mitotic cell death, or cellular consequences that arise from mitosis without cell division. So if cytokinesis doesn't take place, cells have to recognize that they are in a polyploidy, in a tetraploidic state, and have to respond appropriately. Finally, we'll have 3 more talks covering aspects of requirements for entering mitosis and meiosis. And one of these talks might also be interested for listeners to session number 3, because there we'll discuss the role of an RNA processing protein that controls gene expression required for cell proliferation. And finally, we'll have one talk that connects cell cycle progression and cell differentiation. And you will see a lot of movies like the one that I could show here from Marin Barisic, my co-chair, who is in Copenhagen. And we will have a technical presentation connected to this session, which will introduce a lattice light sheet microscopy from ZEISS. And now I would like to hand over to Marin Barisic in Copenhagen, who will introduce the 3 main speakers of our session. Marin, please.
Hello, everyone. It's my great pleasure to be part of ÖGMBT meeting once again. So last time I've been participating during my PhD in Innsbruck some years ago. So as Stefan said, our session is on cell proliferation and cell division, and we have 3 fantastic main speakers that will cover topics as mitotic spindle function, mechanobiology of mitotic spindle, and spindle assembly checkpoint. So our first main speaker will be Iva Tolic. Iva Tolic has obtained her PhD at University of Zagreb, with work being partially done at Harvard University. Afterwards, she moved first to Denmark and later to Italy to do her postdocs. And in 2004, she started her own research group at Max Planck Institute in Dresden, in Germany. After 9 years there, she came back to Croatia and founded her research group at Ruđer Bošković Institute. She is mainly interested in biophysics of the spindle in mitosis and meiosis, as well as in functions of microtubules and motor proteins. And she will be giving a talk on mechanobiology of the mitotic spindle. Our second main speaker is Helder Maiato from Porto in Portugal. He obtained his PhD in 2003 in Porto. He went to the PhD was partially done with Edinburgh University, and then he did his short postdoc in Albany, New York State. After that, in 2005, he started his group in Porto at the Institute for Research and Innovation in Health, I3S. And his main research interest is the spatiotemporal control of chromosome segregation during cell division. He will be giving a talk on mechanism of kinetochore fiber maturation. Our third main speaker is Sabine Ello. She obtained her PhD in Toronto in Canada, and she spent some years as a postdoc at the Max Planck Institute in Munich afterwards. In 2010, she moved back to Canada, where she founded her own group, and her main interest is studying the function and regulation of mitotic kinases with special interest in spindle assembly checkpoint. She will be talking about regulation of mitotic phosphatase activity during cell division. In addition to our 3 main speakers, as Stefan said, we will also have 8 short talks by young scientists, which will cover topics as chromosome congression during pro-metaphase to metaphase transition, spindle assembly checkpoint regulation, regulation of meiotic entry, as well as centrosome signaling and proliferation control. So I hope to see many of you next week during this very interesting session. Thank you very much.
Thanks a lot, Marin and Stefan. So we move on to session number 8 on toxicology, which will be also played by Zrinka. I think this is also again pre-recorded.
Yes.
Good afternoon. At this year's ÖGMBT annual meeting, we have for the very first time a session fully dedicated to toxicology. Please save the date on the screen. Now, modern toxicology is a highly integrative discipline and requires an in-depth understanding of chemistry and molecular processes, as well as a knowledge of sensitive and reliable detection methods. As such, we have an exciting and varied session in store for you. This session will cover all aspects of toxicology, including multidisciplinary approaches and cellular, molecular, and analytical techniques, methods to assess exposure, and mechanisms of toxicity associated with chemicals related to human, animal, and environmental health. The session will be chaired by Joanna Gossner from the Institute of Medical Biochemistry and Herbert Oberacher from the Institute of Legal Medicine at the Medical University of Innsbruck. The key speakers will be Saskia Trump, who is Deputy Head of the Molecular Epidemiology Department at the Charité in Berlin. She will speak about a highly pertinent topic, namely single-cell transcriptomics as a tool for therapeutic target identification in COVID-19. Then we have Johannes Kirchmeier, who is a professor at the Department of Pharmaceutical Sciences at the University of Vienna and who is an expert in cheminformatics. He will be talking about computational prediction of the metabolic fate of small molecules. Simona Moser from the Department of Pharmacy at the Center for Drug Research at the University of Munich, whose background is in chemistry with a particular emphasis in natural product research. She will be enlightening us on the topic of phyllobilins. These are ubiquitous natural products with pharmacologically relevant activities. The session will be concluded by Herbert Oberacher from the Institute of Legal Medicine here at the Medical University of Innsbruck. He is going to be informing us about the possibilities and limitations of non-target LC-MS in toxicology. In addition, please don't miss out on the exciting series of flash talks and short presentations from several budding toxicology enthusiasts. Thank you for your attention.
Thank you very much. So we move on to the next session, which is session number 9, single-cell multi-omics. Francesca?
Hello, I will now share my slides. Okay, here you are. So, hello everyone. It's a great pleasure to invite you to participate in our single-cell multi-omics section that will take place on Wednesday, September 29. Just a few words to introduce myself and my co-chair, Andreas Pirker, who unfortunately couldn't participate today. So my name is Francesca Finotello, and my research focuses on the computational analysis of bulk and single-cell multi-omics to investigate tumor immune cell interactions. And after several years of exciting research in the lab of Zlatko Trajanowski, I have just started my own lab at the University of Innsbruck. And Andreas Pirker is a physician scientist at the Medical University of Innsbruck, focusing on translational research. And his work aims at elucidating the composition of the tumor microenvironment, especially in non-small cell lung cancer. And we propose these session topics because single-cell technologies have opened completely new avenues to study molecular processes by generating vast amount of sequencing data from the fundamental units of life, which are the cells. So, for example, computational analysis of single-cell RNA-seq data allows the characterization of cell types, cell states, and differentiation trajectories based on the similarity of the single cell transcriptomes. But now new emerging technologies also allows to generate multimodal data from the same single cells, including genomics, epigenomics, transcriptomics, and proteomics data. And we have a fantastic lineup of speakers presenting 6 short talks plus 2 plenary lectures. And we also have a company presentation and a breakout room event by 10x Genomics. So our first plenary speaker, Christoph Bock, is a principal investigator at the SEM and professor of biomedical informatics at the Medical University of Vienna. And in his talk, he will guide us through an exciting journey showing how the combination of top-notch experimental and bioinformatics methodologies can shed light onto the epigenetic landscape of cells, so to bridge their past histories with their future potential for adaptation. Our second plenary speaker, Dieter Lambrechts, is a full professor at the University of Leuven, Belgium, and a group leader and science director of the VIB Center for Cancer Biology. And in his talk, he will present an amazing single-cell multi-omics studies in breast cancer patients treated with immunotherapy, where his team reconstructed a dynamic map of intratumoral changes underlying patient response to therapy. So please mark September 29th on your calendar and don't miss this great opportunity to learn more about single-cell multi-omics research. And thank you for this opportunity. I will hand now back to Eso.
Thank you very much. So we move on to session number 10 on metabolic signaling, which will be again a pre-recorded presentation.
Session 10 of our conference focuses on metabolic signaling. My name is Katrien Tediek, and I will chair the session together with Ulrike Rehbein from the Lab for Metabolic Signaling at the Institute of Biochemistry of Innsbruck University and with Katrin Batschinger from the Institute of Biological Chemistry at the Medical University of Innsbruck. We will focus on mechanisms of nutrient sensing and signal transduction that control metabolic homeostasis, and it is our particular aim to bring together physician scientists and basic researchers in order to discuss and find novel ways to translate our findings into the clinic. Our keynote speaker is Lisa Petry-Henske, professor of medicine at Harvard Medical School, and she is also a practicing medical oncologist at the Dana-Farber Cancer Institute in the United States. Lisa is also director of the Center for Lung Research and Clinical Care at the Bryan and Women's Hospital in Boston. She and her lab discovered that lymphangioliomyomatosis, or short LAM, is caused by mutations in the TSC1 and TSC2 genes. And till today, her research focuses on cellular, metabolic, and immunologic mechanisms underlying tuberous sclerosis complex disease and LAM pathology. In her lecture, Lisa will present her latest findings and future perspectives in this field. Our two plenary speakers are Marit Westerterp and Christiane Opitz. Marit Westerterp from the University Medical Center Groningen in the Netherlands will speak about T-cell cholesterol efflux pathways in aging and atherosclerosis. And Christiane Opitz, clinician scientist from the German Cancer Research Center in Heidelberg, will speak about metabolic signaling through the aryl hydrocarbon receptor in cancer. Our session spans exciting topics of metabolic signaling in health and disease, and in particular, I would like to highlight the talks.
Ja, aber die kan do the video now. Sorry.
Okay, yes, so sorry, we can continue now. With
we continue, okay, good.
Yes,
so
we continue.
We move on to session number 11, which is biotech session from Georg Casari and Christoph Griesbeck.
Hello, everybody. I would like to invite you to the session number 11, which is going to take place on next Thursday, September 30th, in the afternoon at 1:15 in the afternoon. I'll just share my screen. This session will be chaired by Christoph Griesbeck, who is associated to the MCI, the Entrepreneurial School of Innsbruck, and myself as a representation of the Biotech Austria and Haplogen, a company. We have set up this session as a collaboration between the ÖGMBT and the Biotech Austria Association to give a good insight into what is actually happening if an invention or an observation goes into practice and enters society. So I would say the theme of this session is bringing fruits of research and discoveries into use by our society. We will report on academic discoveries that are very close to be used in applications in companies or in sort of applied sciences. But also we will show you examples companies will present that have done the step and translate research and discoveries into a service, a product, and are entering the market and serve our society and in some cases patients or other needs. We will also show you a breadth of where biotechnology is actually active that starts from medicine and medical applications and topics through environmental topics down to material sciences. So you will see that the breadth of biotech and the impact of biotech is very broad. Our session will be organized in 7 short talks, and we will have 5 flash talks. So it's a packed program with short, informative talks and presentations. The final, we will end our session with a concluding keynote presentation that will be given by Josef Penninger, that probably everybody knows, who is now associated to the University of British Columbia in Canada. He will talk about a discovery that he did in his academic research many years ago on a very, very different, researching in a very, very different area that has now recently become particularly important and gained a lot of focus and probably also a lot of medical relevance with our pandemic that is coming through COVID. Yes, I hope that you will join us at our session on Thursday next week. It is meant to give you some idea about what can be done if you want to enter the next step, do the next step, and enter from the research and science into bringing it to the market or bringing it to the patient. Thank you very much, and I hope to see you there.
Thank you, Georg. So we move on to session number 12. The chairs are there, myself and Rüdiger Schweigreiter. Rüdiger, maybe you would also like to introduce yourself while I'm sharing the screen. Rüdiger? I don't know whether maybe he's not there with us. So then I will present the session myself. So we have here a session on the mechanisms of neurodegeneration that's going to take place on the 12th of October. So we have 2 excellent speakers, plenary speakers, Tiago Otero from Göttingen in Germany and Per Nilsson from the Karolinska Institute in Stockholm, Sweden. Tiago will tell us about glycation, which is not so well known post-translation protein modification, and he will talk about the role of this glycation in Parkinson's disease. Then Per Nilsson, he is a relatively young group leader who just started his laboratory 3 years ago at the Karolinska Institute in Sweden, and he will talk about autophagy in Alzheimer's disease. In addition to that, we will have 6 short talks and 5 flash presentations. And the short talks, as you see here, are from various different countries. So we have tried to make this session as international as possible. So we'll have talks about all different aspects of neurodegeneration, ranging from, again, Parkinson's, Alzheimer, but also frontotemporal dementia, as well as amyotrophic lateral sclerosis, all with the focus on the molecular mechanisms, how neurodegenerative diseases occur and what we can do against them, but all on a cellular and molecular level. In addition, we have, as I said, 5 flash presentations, all from Austrian universities, as well as also from 3 presentations from Italian universities. So overall here we combine 6 different countries into this presentation, which I'm very much looking forward to welcome all of you on the 12th of October in this ÖGMBT meeting. Thank you very much. With this, I stop this presentation and hand over to session number 13, which is stem cells by Frank and Nadine.
Yeah, thanks, Hasso, for handing over. And yeah, I'm here to welcome you to the stem cell session teaser. And actually, I think it's not really necessary to make a teaser for stem cells, because as we all know, without any doubt, these cells are the most fascinating cells on the planet. And we hope that we can convince you within the session that this is really true. My name is Frank Edenhofer. I'm head of the Department of Molecular Biology at the Leopold Franzens University of Innsbruck. And together with my colleague Nadine Ortner from the Department of Pharmacology and Toxicology, we will share this session. I think we compiled a unique program that we hopefully show you how enormous the potential of stem cell application for therapeutic applications, but also disease modeling is in the current research. During the past decades, stem cell emerged as an attractive model to study the pathogenesis of complex diseases, as well as to provide a virtually unlimited source for therapeutic interventions. And this session will aim to give you most recent insight into the potential of the stem cell technology, and in order also to explore the potential to understand pathophysiological mechanisms of human disease. So we are glad that we have 2 very renowned speakers, invited speakers in this session. First, it's Professor Gaia Novarino from the Institute of Science and Technology, Klosterneuburg. Gaia Novarino will give us fascinating insights into her research that aims to systematically investigate genetic defects that result in particular in neurodevelopmental disorders, namely autism spectrum disorders. She's using CRISPR technology, 2D, 3D differentiation organoids, and also metabolomic studies. So that will be an extremely interdisciplinary and fascinating insight into current research of stem cell technology. As a second speaker, we will have Professor Dimitri Kreinch with us from Chicago. And he is actually one of the leaders in the field of modeling Parkinson's disease. His lab successfully used PD-specific stem cells artificially reprogrammed to study pathophysiological dysfunctions. And as you can see from the title, he will give us new insights into the role of mitochondria and lysosomes in Parkinson's disease. This session will be complemented then by 3 short talks from the abstract submitted. These will cover extremely interesting applications from stem cell-derived blastoids, as you can see here by Teresa Sommer from the Vienna Biocenter. So using stem cell-derived models for early human development. Ahmad Salti from the Kepler University in Linz will talk about metabolic dysfunction due to a genetic defect. where stem cells provide a relevant human model to analyze the dysfunction. In the cell culture edition, finally, we will have fascinating insights also into 3D cortical organoids that will be used to better understand early development of the human brain. Yeah, with that, I think we have a very nice program, and please mark your calendar, be ready at October 13 to virtually travel to the stem cell universe, and Nadine Ortner and me look forward to meeting you there. Thank you.
Thanks a lot, Frank. So we move on to session number 14 on host-pathogen interactions. The chair is again myself. I took over the session, so I'm personally not a host-pathogen interaction person, but I took over the session, and I have the pleasure to welcome here two very good speakers. One, a local speaker from Austria, Tim Skern from the Medical University of Vienna, who will be talking about viral proteins and how they are incredibly multitasking in their functions. Then we will have Bernd Schmeck from University of Marburg in Germany, and he will talk about RNA networks and host-pathogen interactions. So this might be also a talk that is of interest to people from the session on RNA biology. In addition to that, we will have 3 short talk presentations from 3 different Austrian universities and 5 flash presentations to round this up. So this session will really combine all aspects of host-pathogen interactions from the cellular level, from the host level, as well as from the pathogen side, and how these two interact with each other. I think this is very timely, this session, especially since we have been hearing Corona all the time. There is also a talk about coronaviruses. You will. I hope that all of you will come and visit this session on the. When is it? On the 13th of October, which is like the parallel session to Frank's stem cell session. With this, I stop my presentation and hand over to the last session, which is session number 15, Translational Oncology. And this is again a pre-recorded session. Okay,
So hello from my side. My name is Heidi Fiel. I'm a molecular biologist from the Department of Obstetrics and Gynecology. My co-chair is Heinz Zoller, an internal specialist from the Department of Medicine 1. We are both from the Medical University here in Innsbruck. So Heinz and me will look forward to welcoming you to our Translational Oncology session and sharing exciting talks with you. At the beginning of the session, we are happy to have a great plenary speaker, Jorge Barbosan, who will talk about the impact of stromal compression on tumor development. So the inhibition of the so-called mechanotransduction is a potentially promising way for future innovative therapies. Afterwards, we will hear in short talks about therapeutic vaccination, STAT5 in anaplastic large cell lymphoma, or the inflammasome in prostate cancer. In the so-called science flashes, we will see in 9 speed presentations new research data on early diagnosis of colorectal cancer, the role of AXL and the TGF-beta signaling in hepatocellular carcinoma. In a prostate cancer focus, there are presentations about cancer development, cancer microenvironment, and therapeutic concepts in prostate cancer. But we will also hear about therapeutic applications of PARP inhibitors in high-grade pediatric solid tumors and gene therapy in primary human glioblastoma. So our second plenary speaker is a fantastic expert in the field of liquid biopsies. So we are happy to have Catherine Alix Banerbier on board. She will talk about technical specifications, clinical applications, and we will talk about the establishment of CTC-derived cell lines in colorectal cancer. And finally, we are proud to announce Douglas Hanahan for the keynote lecture. He will discuss the hallmarks of cancer in the context of translational oncology. In other words, what clinical applications can be derived from these hallmarks. So this keynote lecture should create perspectives and will be the crowning final of this year's presentations at the ÖGMBT meeting. So we hope that we have attracted your interest and that you will join us on the 14th of October. Thank you for your attention.
So with this, I think we are done. We have presented, all chairs presented their sessions and the speakers in these sessions. With this, I hand over to Zrinka, who and Andrea, who know what are the next steps.
Thank you, Hesso, for such a marvelous moderation of the session pitches, and thank you all the chairs for introducing the exciting program we will be having in next 4 weeks. So now I will announce we will take a coffee break, and we are back on Zoom and also live streaming on YouTube from 3:30 on. We have a really engaging program with Life Science Awards Austria 2021, bestowal of the awards. And please come and hear what our awardees have to say and all about their project. So I'll see you back at 3:30. Thank you very much.
Lucas, can you hear me?
It gives you a very nice showcase of the quality. The quality of Austrian research is extraordinary high, and these young investigators are the reason why this research machinery is driven from the bottom to produce excellent science. What is nice then, when you go back for all these years, not only that you see familiar faces, and you come to a statistics section. And we asked the awardees, we talked to them, and 75% observed that these prizes had a positive effect on their career development. They got more attention, they got maybe job offers, and they had an advancement of their career paths. Almost 50% of the awardees received additional funding. And over 80% of the awardees increased their visibility and expanded their network through our society and through the public attention and recognition they got after the prize. On the right side, you see a statistics about the current positions. So there is a large part in academia, but there's a considerable part in business, in institutions, and there's a small part we couldn't trace back what the actual position at the moment is. But from this picture, you see that we cover both sectors, the applied sector and the basic research sector, which is for us very important. Now, having said that about the prizes, I would now and in the following, we will come to the heartpiece of the prize ceremony, and that is that we will first briefly introduce the awardees to you. We start with the research prizes, the research awards. We call the awardees up briefly. We will then hand over the certificate of the prize, the diploma basically. And then we will line up and make a picture. But before we do that, it's my great pleasure to invite now Dr. Unterer from the Federal Ministry of Digital and Economic Affairs for her speech. Please, the stage is yours. Format with such a broad
distinguished president of the award, winning scientists, dear friends of the international life science community. Thank you very much for inviting us to the video.
Take a picture. But before we do that, it's my great pleasure to invite now from the
We readily contribute our funding towards the Austrian Life Winning Science Awards International Life Science Community. Once in Austria, and our ministry carries life sciences in its DNA since the onset of these disciplines having become a key technology and a business in the early 1990s. Since then, our ministry has set up a well-designed mix of incentives to foster the life sciences along the broad range of its value chain. Just think of the program LISA of the AWS, the many programs suitable for life sciences of the FFG or the Christian Doppler Promotion Agency, meanwhile supporting about 1/3 of its labs in the cluster life sciences and for medicine.
Bigger picture. But
Before we do it now, it's my great pleasure to invite science industry, and it would not work better otherwise. So one of our latest babies to support to the community was Wings for Innovation, operating successfully since 2019. But one of our most constant issues has been from the very beginning and still is our support to young talents and tomorrow's leaders, which includes our prizes and awards. So this instrument is the best option to acknowledge scientific and scientifically applied achievements, to give visibility to young scientists, and to focus their careers. And virtually all of them absolutely deserve the distinctions. Beyond that, and for the upcoming annual event, we are happy to have supported a testimonial of former Austrian Life Science Award winners, which the ÖGMBT has marvelously collected in the new flip book. We hope this booklet will also contribute to encourage students and researchers to go for a life science career, and that these ambitions will also result in the provision of our life science location with ambition and well-skilled personnel that we need so badly. To make the story short, I congratulate this year's Life Science Awards awardees, and I'm eager now to hear their interesting talks and their trend-setting stories. Thank you very much for your attention.
Yeah, this testimonial as a document for the Science Awards. Thank you once again to the listening, but I have to extend this by the number of people here. I want to thank the award winners, of course, that they were willing to share their success stories, that we could raise them, but all this was very difficult. We needed to pay the project partners for Biolution. Dr. Iris Grünert is the mastermind behind that, and I'm very happy that she's here, especially since we are former colleagues who worked together already in EPL and Heidelberg. She was the mastermind, and Paul Grünert was compiling the questionnaire and condensed the answers into the text. I was glad with you here. Anuradha Genrich for molding, varying text, photos into one layout, and Daniela Hubusek for her organizational talent, which was instrumental for keeping the timeline. This was a big project that was running in the background. And here I should thank also the ÖGMBT Executive Officer Alexandra Kasitov for having this great idea, initially to generate the showcase, and Andrea Bauer for her competence and for her hard work for taking on the project management and implementation during Alexandra's maternity leave. Thank you to all of you, and a big applause. So now it's my pleasure to announce the first 3 winners of the Life Science Awards Austria category Research Awards. The first category Research Awards is the category Basic Science, and the winner is Domen Compute, and he crystallized a mitochondrial respiratory complex. And please come to the stage that I can hand over the prize to you. Later introduce with a bio-sketch speakers and the scientific topic, but now you just remain here. Please line up. We will now we make afterwards. We make afterwards. Benjamin Salzer is the winner of the category Applied Research, and he had an engineering project for a car therapy, a very interesting new procedure, which enables us to control car function. And you have, please. And the third category is excellence and societal impact. And it goes to Charlotte Zeig. And she has also worked on a car therapy. And with our orally available product, which I think is quite remarkable, and this will have quite societal impact. And you have highly deserved this, and please come out that I can end this beautiful. Now we make the first picture. Please mute the beamer. Mute the beamer, I do that. I promise to do that. Thank you very much. Jetzt die Fotomutter da allein mit dem Gewinner und dann der Gewinner allein. Oder jeweils der. Not just the winners. The winners? Yes. Thank you very much. Now, we come to the scientific lecture of the first awardee of Domen Compute. He is from the IST in close to Marburg. The title is The Coupling Mechanism of Mammalian Respiratory Complex 1. Domen is a structural biologist with a strong interest in membrane protein complexes, metabolism, and signaling. And the challenge for a structural biologist couldn't be higher than looking at membrane proteins, because this requires a special talent to do so on many, many complicated techniques. He completed his PhD at the IST in 2020 to the scientific focus on the structural characteristics for the IST of mitochondrial respiratory chain in His title is The Coupling Mechanism of postdoctoral as fellow at MSc Laboratory of Domen is a structural and there he studies AMPA receptor with a strong interest in membrane protein complex metabolism. And in the junior research fellows at this college at Cambridge, then now you have received remarkable results. So many remarkable inserts. You already completed this PhD, and please come up to the stage. I will launch the presentation for you. Okay. Thank you very much for a nice introduction. It's a big pleasure to be here and to get this. Thank you for engaging this day. So I have 10 minutes to explain you a little bit about my paper, which focuses on the respiratory complex one. So, but let's begin with this figure. So these are all the parts of the chemical reaction happen. ISD coming into science where we focus all the structural characteristics for the ISD of mitochondrial response between so I have 10 minutes to explain you a little bit about my paper. The way this system works is that the outer memory. But let's begin with this figure. And these are all the part of the chemical reactions that we have on the ISD coming into science where we focus all the structural characteristics for the ISD of mitochondrial response between so I have 10 minutes to explain about the way this system works focuses on complex 1. So complex 1 is the first of these enzymes and the largest one, and it has remained, the structure has remained elusive for much longer than some of the other components of respiratory. So it's composed, it has 1 megadalton inside, and it's in nano, it's composed of 45 subunits. So 14 of them are colored here. There's the 4 subunits which catalyze the reaction. Then there's 31 supernumerary subunits which help with regulation and assembly demands. And then on the top, out of the membrane, we have the peripheral arm, which is kind of the electron transfer reaction center. So the NADH, the first substrate binds here, electrons get transferred along the chain of iron sulfur clusters, and then at the bottom, we don't accept them, and the reaction proceeds to glycolysis. And then in the membrane, we have the membrane arm middle. This is the proton pumping arm. So the protons are led through the 4 separate proton pumps here, which provide the proton brain. Now, we don't know how these two reactions are coupled. How can something that happens here provide transmit the energy of the protons? So to do this, we decided to study the sheet mitochondrial complex 1. We start, and sheet heart is very rich in mitochondria. So we purify complex 1 from sheet hearts. Put it on the prion grid, and then analyze tens of thousands of images that we got. We made a little trick, which was to incubate complex 1 with substrates. You start the reaction, and the reaction was proceeding while we froze the sample. So, and then we could, with 3D reconstruction classification approaches, separate the different reaction intermediates. So we also collected a bunch of other data sets with one or the other substrate in the inhibited state to really understand what is going on. And the resolutions that we achieved were quite nice for some of these conditions, up to 2.3 angstroms. Just to show you what we can see in these resolutions, we can see lipids, mitochondria specific lipid. We can see the substrates very clearly. We can define the positions of the atoms. We can see the cofactors, such as cluster clusters. We can even see translational modifications. and acylated and methylated residues, and most importantly for the mechanism, it's the water molecules. So water molecules are the ones here, which are the wires and the proteins through the complex. So what we could see immediately from our analysis was that complex 1 exists in two different formations. One is the open structure, and the other is the closed. So yeah, so this would be the closed structure with the two arms close to each other and open up as well. We could immediately see that the turnover is catalyzed in this open. And then if we look closely where the quinone binds, so we use the electron acceptor substrate, you can see that in the closed state we have quinone bound deep in the pocket where it can receive electrons from the other side of the cluster. In the open state is bound very far away from the other side of the cluster. This was the next clue for the mechanism. Look at the whole binding site, right? The closed state, the binding site is very narrow, very elongated. The quinone can accept the electrons. And then once the complex opens, the whole cavity opens up as well. Quinone is pushed out, and water molecules can enter into this cavity. Then when we have extremely reduced complex, just pH, we have even further opening and closing of the top part of the cavity. So maybe to make it clear in the video, this would be the closed conformation of quinone. So the blue molecule is the quinone. Then once the complex one opens, you see the quinone travels back down. It is pushed by all of these mobile loops in the binding. So, but we still don't know what drives the openness, right? So we know how the quinone gets reduced and how it comes out in and out of the pocket, but we don't know how to drive the proton. So when we look, so this is the quinone cavity binding site here, and it's connected by a string or a wire of water molecules all over the membrane complex. So and these would be the three separate antiquarter units which run the protons across the membrane. Now, when we analyze the positions of water molecules, the residues which come from this, we have a big distinction between the open and the closed conformations. So in the open conformation, this position are the green arrow. We have a helix which is basically breaking the water wire, and there is no communication between the quinone cavity and the rest of the proton. Whereas if we look at the closed conformation, the helix moves out of the way and allows the water wire to form fully and for there to be communication between the membrane and the so maybe to make it clearer in the video, we have open and closing of the complex one. And if we zoom in on this helix, shown in pink here, we see this extreme rotation of the tyrosine residues here, which basically opens up the channel for water molecules to flow through and initiate the protein. So I think this is it with my 10 minutes. I would like to acknowledge my professor, Lipid Sazanov, who was my thesis supervisor during this work. I would like to acknowledge the public sources, facilities were collected, analyzed the data, and all of my former colleagues who were wonderful to work with. Thank you very much.
Thank you very much for this masterpiece of structural biology. It's an enlightening structure of, it looks like a beast, this complex. I thought, it looks like a beast, frightening, you have courage and the vigors of congratulations. The paper very well established science.
Now, I will launch the second presentation that I can introduce our next speaker. Now, the next award goes to the category Applied Research, as we said before, and this is Benjamin Salazar. He comes from the St. Alba Children's Cancer Research Institute here in Vienna. And Benjamin Salazar studied medical biotechnology at the University of Natural Resources and Life Sciences in Vienna, and did a PhD in biomolecular technology of proteins. Presently, he is a postdoctoral fellow in the Christian Doppler Laboratory for next generation of CAR T cells. So that's not a surprise because he did groundbreaking work in CAR T cells at the Children's Cancer Research Institute in Vienna. His research interests focus on cancer immune therapy using CAR T cells, and in his work is harnessing the tools of synthetic biology and protein engineering to enhance the therapeutic potential. Anyway, we are all looking forward to your presentation.
Thanks, Mr. Eric, for an introduction. I'm also very honored to be here today, and I would also like to thank you again to see what happened today. So, in the next 10 minutes, I would like to present to you our study we published last year with the title Engineering Inherited CARs for Combinatorial Antigen Recognition and Reversible Control of CAR Function. So, first of all, what is a CAR? So, a CAR is a chimeric antigen receptor, and to put it shortly, it's basically a new class of synthetic immune receptors. And here you can see the basic structure of these CARs as they are expressed in human T cells. It's a very important class of immune cells. And in fact, these CARs consist of two parts. So first of all, the antigen binding domain top, which gives the CAR its functional specificity, and you combine these domains with signaling moieties derived from T cell receptor, or short TCR. In fact, TCRs are quite big. You don't think of the protein, but just the intracellular part. So the CD3 domain, which is enough to give the CAR its function. What can this receptor do? Basically enables the MHC-independent retargeting of T cells and tumor cells. So that means by just selecting the right anchor here, you can basically redirect the T cells to virtually any tumor entity. And that has been done very successfully in the past for certain kinds of leukemia and lymphoma, where you just target CD19, which is the marker which is expressive of all B cells. This first generation CAR was in iterative cycles improved to yield second and third generation CARs, which just differ in the composition of this intracellular part because they have more functions. So the kind of therapy works very well in certain types of leukemias, but still we need better control of the cells because we still have huge adverse events which can be fatal, and we can't control the cells very well in patients. That's why in my study we wanted to tune these receptors in a way to introduce novel functions and novel control mechanisms. And to put it more precisely, we wanted to, on the one hand, to create a sort of on switch to very precisely control the activity of these cells in the human body, to remotely control these cells. And on the other hand, we wanted to achieve something which is called combinatorial antigen recognition, or NCATES, for increased specificity, but I'll come to that in a minute. So what is very important to mention here is that CARs, as we know, that these molecules, they are not present in a strictly monomeric form, although they are depicted like that in most applications, but they are to some degree present in a dimeric form. And that has been shown to be caused by dimerization of both existing residues in the stored region of CD alpha and CD28, so molecules, part of molecules which have been used for CAR engineering. And this clustering of dimerization has been shown to be positive of oligomerization, tonic signaling, and exhaustion of CARs, and eventually failure of this kind of therapy. So that's why we propose to use this phenomenon to do something useful with it and to create a novel platform of so-called ability control CAR T cells or short vectors. How does it look like? So just imagine a normal high-ability CAR which is currently used in clinics to treat patients. You can appreciate, so this CAR has high speed. So you can appreciate that when this CAR binds one single antigen, so monovalent bond is formed, and the lifetime of this interaction is very, very strong and very, very long, because and the affinity is too high. Thus, the T cell will be fully triggerable only if one million bonus bonds. But if you would not take the same card, it would reduce the binding affinity below certain thresholds. This monovalent bond won't be enough to trigger the T cell because the lifetime is too short, so the T cell just falls off the tumor cell. But if the same card is able to bind two antigens at the same time, and on top of affinity, a certain contribution of ability, which is the cumulative strength of multiple affinities. So basically, we pump up the effective affinity and binding strength by bringing two binding sites together. And I just want to mention here that this effect is not just additive, but rather multiplicative. So we really get a huge impact on the effective affinity. Now, until now, it's not possible to exploit and control it in a precise manner. Thus, in order to exploit the ability for cars, we propose to include three key design principles into car engineering. First, the usage of low affinity antigen binding domains, as already introduced. Present on a monomerized car vectors, we do not want any form of unwanted dimerization, but we would include an additional domain into these cars or to dimerize these cars later on. So we want to induce very precise dimerization of these molecules only when we want to. In that case, we will have a platform which is dependent on two signals, a small molecule dimerizer, so the on switch, if you want to put it like that, as well as the presence of an antigen on the tumor cell. In that case, we'll have the first application of our platform on switches. For increased specificity, we wanted to build something which is called an APIs. So just imagine the same platform as on the previous slide, but now with two different car chains. We have two chains which are quite similar, but they have different antigen binding domains. We can target two different antigen binding antigens. And both present on a monomerized car backbone, and now with a gate dimerization system. So we want to very precisely control the dimerization of these two chains. In that case, we have three input signals. Present on the small molecule dimerizer driving dimerization, as well as a combination of antigen A and B on the tumor cell. So here we are really dependent on two input signals, two antigens on the tumor cell. And with that, we can vastly improve the specificity of this therapy because we can be more selective for the tumor cells and not target any tissue. So with that, we wanted to see whether it's feasible to control cars by ability. And for that, we wanted to see whether it's feasible to switch cars on and off. For that, we used following car architecture, as already introduced. We used an antigen binding domain, which is called SSO7B, which binds with low affinity towards human EGFR as our own antigen. And this is present again on a monomerized car backbone. And as a dimerization domain, we use the so-called FK12 system, which can be homodimerized very efficiently with a small molecule called AP2187. As a model system, we used an NSG mouse model, so that in vivo work, where we injected the leukemic cell line called M6, expressing C90 as our benchmark, as well as human EGFR as our model antigen. And here you can see the outgrowth of these cells over 11 days. So here you can see the signal of the tumor cells in each mouse. First, we confirmed that the CD19 CAR, which is very used in the clinics, was very, very potent in controlling outgrowth. The same was true when we used the high affinity version of this CAR to serve as a positive control for this specificity. But when we used the extra CAR, so low affinity, but in an off state, so no dimers are present, we lost this tumor control. But very important and very interesting, very important to see was when we induced dimerization by giving this small molecule drug, we were able to restore the activity of these cells to a level which was comparable to benchmark controls. So at the monistic view on things, we also repeated this experiment over a long time frame and looked at the survival of these mice over 40 days. Here I want to highlight two important things, messages. First, that the avid kind of off state, so the dotted red line, was basically indistinguishable from T cells that were not treated previously. So there's no background, which is nice. And that the avid kind of on state, so the full red line, was very similar to our benchmark controls, so C19 and high affinity version. So that showed us that conditional dimerization enables the remote control of cellular function. For the second application, the avid CAR, so for increased specificity, we had to twist the system a bit So as already introduced, we used two different CAR chains, binding to either EGFR or HER2. These are our two model antigens in that scenario. And as a dimerization domain, we did not use a conditional one, so not switchable, but rather a sensitive one, because we did not have any good dimerization system at that time. And so this is a zipper pair which forms a very, very stable dimer, no matter what, so it's not conditional. As a model system, we used a quite sophisticated MSG mouse model again, where we injected into each mouse 4 different populations of this leukemic cell line. So these are cells which are expressing a low antigen, only HER2, only EGFR, or the combination of both. And this is a combination. We injected them in a one-to-one picture into each mouse. Later on, we treated these mice either with T cells as an active control, a high affinity control part, a positive control, or our actual bispecific part. 30 days later, we took bone marrow samples of these mice and then tested for the outgrowth of all these 4 populations. We wanted to see whether there's a change in outgrowth of different populations. So to give you an idea how it looked like, here's the flow cytometric data on day 13. First, after the treatment with T cells, so you can appreciate that we still have all 4 populations present as expected, although the output kinetics were a little bit different. When we use the positive controls, so a high affinity EGFR specific card, as expected, we lost all cells expressing EGFR on the right-hand side of the plot, because it's not very selective. But when we use the actual bispecific EBIT card, we were very happy to see that we very selectively get only cells expressing both antigens here in the right top corner, while the single positive controls were still there. To give the whole plot of numbers, we quantified the cells per organ, so each dot represents 1 mouse. So the bone marrow on day 13, and here you can see the quantification of either MAPC cells without antigen, with both, or the single positive controls, or the T cells themselves. So again, 2 very important messages. First, that the EBIT CAR, the high affinity version of this CAR, so here shown in green, it was very good in killing all cells expressing EGFR, so these 2 plots or columns, while the bispecific EBIT CAR was very good in selectively killing only cells which have both antigens present, while the single positive controls were spared when you compare it to T cells. So it was very, very specific. So that we could show that any EBIT CAR is able to specific targeting of this antigen combination, EGFR as well as HER2, also in With that, I would like to summarize. So I showed you today that EBIT CARs are able to integrate reducible as well as logic control functions, so sponge character as well as combinatorial antigen recognition into CAR T cells by using the DT effect. This can only be achieved by introducing key design principles into our engineering. This being controlled dimerization as well as the usage of all the platform is very versatile. We can build different things, and just to mention that we can build features, and gates, as well as gates for membranes as well as some antigens. I could not cover everything today. If you're interested, just have a look at the paper. So that like a lot of people involved in studying. One foremost, it's another laboratory for next generation CAR T cells. So my supervisor Martin here, as well as Charlotte, the next talk, and also all the other people involved in the study of antibodies and our industrial partner in biotech. And you pay attention.
Thank you very much for a very clear presentation. Also, this groundbreaking work at the end, treating patients found a way to the very German Nature Communications was published and was recognized by the community by the jury. It is quite remarkable that now we move on to the second category, but we stay thematically tightly attached. It comes again from the Children's Hospital, the Cancer Research there. And the prize winner is Charlotte Zeig. And she also worked in CAR T cells. And let me just give you a brief introduction. So She studied, let me start it. This paper, this work is also very, very important for the safety, for the controlling of CAR T-cell activation. This is a problem. The principle is extremely well worked out. It works very nicely for some leukemias, but it's always a problem to control it. That when you play with the immune system, it can shoot back. And this is her major contribution. And her paper was also in a very prestigious journal. It was in PNAS and the Proceedings of the National Academy of Sciences of the United States. It has a great importance for CAR T-cell therapies. And the trick that she has used is that she can control that with an orally applicable drug, a new molecular system that can control these CAR T-cells. And this remarkable work she will introduce now personally to us. Walter, have you launched the presentation or should I launch it?
Please launch it, yeah.
Thank you. Please, Charlotte, come to the stage. We are all looking very much forward to the presentation.
Okay, thank you very much for the introduction. My name is Charlotte Seitz. It's a great honor to be here today and to present our work on the confirmation-specific on-switch for the control of CAR T-cells. Currently, I'm employed in the Christian Doppler Laboratory of Next Generation CAR T-cells at Open University, but I did my PhD thesis at the CCRI, so this is part of the PhD thesis. And I would like to briefly reintroduce CAR T-cell therapy. My colleague Benjamin already gave an excellent introduction. So what is the amazing thing about CAR T-cell therapy? For cancer patients, they are treated with their own immune cells. So basically, blood is collected from the patients, and then we can isolate the T cells and genetically engineer them ex vivo. So this means we basically train the T cells to recognize and fight tumor cells. Once they are engineered, they express the so-called CARs on their surface, and they are then called CAR T cells. And we can enrich these CAR T cells and give them back to the patients. So basically, the patients are treated with their own immune cells, just better ones. How does a CAR look like? So it's basically a receptor like a Lego receptor, which just assembles different parts which are already there in nature. So these parts are basically from the T cell receptor complex, and they are important for T cell signaling and co-stimulation. We have a transmembrane domain, and then we have this extracellular portion of the CAR, which is usually an scFv. And this scFv can detect cancer antigens, for example, CD19 or EGFR, and then can bind to the tumor cell, and afterwards we have downstream signaling, and the T cell can then hopefully lyse the tumor cell. As my colleague have already introduced, the CAR T cells are approved, it's an FDA-approved drug, also approved in Europe, and it has been working very impressively in many clinical trials. However, there are certain toxicities which are very adverse. And here you can find some trials which are summarized. So these are different trials, very large trials where all patients have very promising remission rates. But however, you can also see that in all of these trials, different kinds of side effects occur. So these are, for example, cytokine release syndrome, meaning a massive amount of cytokine release, or neurotoxicity, and these can be really life-threatening. So many of these patients go into a coma, and it's really life-threatening for them. Another trial also highlights that in all of these trials, there are some kind of side effects occurring, and this somehow underlines the necessity for finding ways to better control these CAR T cells. So therefore, we have the hypothesis to engineer an on-switch, which we can use to turn these CAR T cells on and off after they have been given to the patient. And for this, we use the protein and a small molecule drug. And the combining of this small drug, we hope to have a conformational switch, and then we can engineer a second protein to recognize this compound-specific conformation. So basically, we engineer this protein and have protein-protein interaction only when the small drug is present, but not when it's absent. And as the first protein, we use human retinol binding proteins. That's a protein that every one of us has in their body. And as the name already suggests, it normally transports retinol. It has this beta barrel shape, and you can see retinol, the blue light here. It's crystal structure, which has been solved previously. And then there is also a synthetic drug, A1120. And when this ligand is bound, the conformation up here in this loop region is a little bit different. So there is a small conformation switch, which the protein performs upon compound binding. And we thought, okay, let's take this drug, which has been investigated for diabetes, so for a completely different incidence and different disease, and see if we can engineer a protein which is specifically only binding to this certain loop conformation. And as the second protein, we use the SSO7B protein, which is a small DNA binding protein from supergroups. So how is this done? We use yeast display in our laboratory. With yeast display, you can express any type of protein on the surface of yeast, flanked by 2 tags. And here, for example, when we have the SSO7B protein, this is not only one protein, but a library with different types of proteins. So in each of these positions, which are highlighted in blue, you have different amino acids, and there we have a huge variety and diversity of proteins. And upon binding of this compound-loaded other protein, the RBP4, we can then select for those proteins which only bind in the presence of the compound, but not in the absence. And this library we already had in hand, so this was actually by Michael Taxmeyer at the MIT. And we started off selecting, and after several selection rounds, we finally saw or found different binders. So binders are always these versions of proteins, which only bound in the presence of the small molecule drug, but not in the absence. So here you can always see increasing amounts or concentrations of the RBP4. So in both cases, the protein is present, but only when we add the drug, we can see a binding signal. And we analyzed this not only like yeast display, but we also analyzed it with other methods, for example, surface plasmon resonance. And here you can again see the binding, the interaction between these 2 proteins in the presence of the drug. And here you can, as you see, the binding curve with increasing concentration, and we can fit this data to a low nanomolar KT value. Whereas with the same concentrations in the absence of the drug, you do not get any signal. And we also additionally analyzed the interaction between the small molecule and the protein, and also got here a nice dose response curve. So to sum this up, we think that the on-switch is very efficient, and we have different versions of these proteins that are up to 500-fold difference between the presence and the absence of the small molecule drug. So it's really specific switch, and can be precisely controlled with this specific orally available drug. Of course, we were also interested in understanding why it's so specific. So therefore, we performed crystallization of this on-switch complex and solved the crystal structure. You can find the details down here. Here again, we can see the RBP4 bound to the A1120 in pink and the engineered protein up here. And what is quite remarkable is that when we zoom in, we see that the engineered protein interacts with the switching loop regions in yellow, but does not directly bind to the compound. So the reason why it's so specific is really because the switching loop regions are recognized, and not because the compound itself is binding to the second protein. And if we overlay crystal structures with the natural form of the protein, the retinol-bound version, here in blue, and then our complex RBP4, you can see that the loops are usually in a place where they would sterically clash with the engineered binders. So that's the reason why our on-switch is so specific and really confirmation specific. Lastly, of course, we wanted to integrate this into a CAR. So here we split the CAR into chains. The one chain was expressed by the T cell on the surface of the T cell containing this engineered protein. The other chain was expressed solidly by the T cells themselves and contained the RBP4. And then we had an anti-CD19 scFv, which is also used in the clinically approved CAR T cell therapies. So upon addition of the small molecule drug, we would then expect the interaction of these 2 chains and assembly of this whole complex and CAR T cell signaling. Looking here at always in gray, the absence of the small molecule drug versus presence of small molecule drug, we have different conditions. So conditions where the T cells do not express any CAR, or they only express chain 1 of the CAR, and you can see that only when they express both chains and we add the small molecule drug, we can get an increase in tumor cell lysis. And this is significant not as high as the control CD19 CAR, but there is a significant difference. And of course, CD19 negative cells were spared as expected. So it's really specific for CD19 positive tumor cells, only when they add the small molecule. When you look at CAR T cell signaling using a reporter cell line, we see the same picture, maybe it's a bit clearer. So only when we have two chains present and we add a small molecule drug, we can see an increase in CAR T cell downstream signaling. So to sum this up, we have engineered this on switch, which is highly specific for ADN20, this already available drug. And we also tested a range of other drugs which are known to bind to protein and potentially would also be able to trigger the switch, but we see that none of these drugs was able to use binding, only again our ADN20 control drug. Secondly, we performed structural analysis and thereby revealed why, or the mechanism of this confirmation of specificity, and integrated this on switch into CAR T cells and used it to control CAR T cell signaling. And this is just again to show you also that in CAR T cells in vitro we can get a nice dose response curve when we titrate the small compound. So potentially later on, when we use this in a patient, we would hope that you can give varying amounts of this small molecule drug, and then also control how active the CAR T cells are in the patient's body. Yes, and with this, of course, I would like to thank especially Benjamin Zeitzer, my colleague during the PhD studies, and my mentors, Lena and Michael Taxenmeier, the OCD Lab, and our funding sources. Thank you for your attention.
Thank you very much for this excellent talk, a very elegant procedure that you developed that will be very helpful, and great recognition of the research that Santana and that Boku and that the Christian Doctor that we can acknowledge to exceptional young scientists from this research. Congratulations to all of you. Now, it's my pleasure to move on to the next categories, and these are PhD awards. And here we go. We have two exceptional young PhD students that get awarded for their excellent work they did during their PhD. And this is Stefan Deleczki-Zaniewicz and Barbara Bachmann. The first category is the category of basic science. And winner is Stefan Deleczki-Zaniewicz, and I ask you to come to the stage, and you come together with a sponsor, a long-lasting sponsor of this category of awards, THP Medical Products. Chantal, please come and join in here for the ceremony, please.
And Lucas, you know our long-lasting
This is
check
the
that we have
long-lasting
here
check.
for many years.
We make them the picture together.
Yes, please mute the beamer.
I mute the beamer, sorry, you should have complained earlier. So I will come here to you next slide. Please stay here. We come to the second winner, Barbara Bachmann, in the category Applied Research. Please. The sponsor is our pioneer founder of biotechnology in Austria, Hollywood, Mexico. It's always a great pleasure to have you here. Sorry, I've not checked, but I can see the picture. And then we move to the middle. We make the picture together, and afterwards we make a picture of all. The ministry sponsored the doctor, the THP, and we do sponsor one all together. Eine Spitze. Eine Spitze. Very good. Eine kleine. Einmal now, I will briefly introduce the first speaker to you. I will activate the screen again and launch the presentation. So, now Stefan Telechki-Zaniewicz studied molecular biotechnology at the University of Applied Sciences in Vienna. And during his PhD studies, he investigated oncogenic mechanisms of leukinogenic fusion proteins in the group of Professor Gavian. And he made the discovery that a family of nucleoporin 98 fusion proteins are involved in biomolecular condensation, which is an important mechanism in leukemia development. Recently, he's working at the St. Anna Research Institute, surprise, and he's interested in another family of oncogenic fusion proteins leading to sarcoma and is working there. But now we want to hear about your discoveries you made during the thesis. The stage is yours. We are very much looking forward to your presentation. Please.
Thank you very much for the introduction, and also thank you very much to the organizers and the ÖGMBT for inviting me and giving me the chance to present this very exciting work that I conducted during my PhD studies, mainly at the University of Veterinary Medicine in Vienna, but also in collaboration with the St. Anna Science Research Center in Vienna. So today I want to tell you the story about NAP98, nucleoporin 98 fusion proteins, and why they are very important in the term of acute leukemia in biomolecular condensation. So to start, very basic, oncogene, so we can have a gene X that has a mutational change, usually it's a gain or loss of function, which leads to a phenotype. A different type of mutagenic event can be a so-called chromosomal translocation that leads to a novel fusion protein that leads to, or that can have different novel functions that may lead to cancer. This has been shown to be exquisite drug targets in the past, and that's why we now investigate type of oncogenic fusion protein. In particular, a certain subgroup of such translocations are multi-partner translocation families. In this, we have the N-terminus of a gene A maintained, which can then fuse to a variety of different fusion partners. And such example of a family is the nucleoporin, the NAP98 translocation family. It consists of more than 25 different members, and it's a very severe disease phenotype in acute myeloid leukemia. In this study, I investigated 5 different of those family members that all maintain the NAP98 N-terminus, which is basically 2 sets of phenylalanine glycine repeats fused to different C-terminal fusion partners. And we express those different fusion proteins in the human leukemia cell line and image their localization. We can see that this is the nucleus in green. In this dotted speckled imaging, we can see our fusion proteins expressed. On top of that, we performed affinity purification coupled to mass spectrometry, where we basically find all the interaction partners of one of those fusion proteins, and we plot them all together. So we have here our fusion proteins, and here in the middle, which I want to highlight, is basically the conserved interactome of those different fusion proteins, which gives the big indication that there is apparently a common mechanism that all those fusion proteins that lead to leukemia share. And if we have a close look at those roughly 160 different core interactors, In terms of biomolecular condensation, we analyzed it in other ways, of course, as well, but this speculative confirmation and then together our analysis gave an indication that there might be something going on with biomolecular condensation. And here we use the p-score, which is a prediction of how likely it is that certain proteins are found in such condensates. And you can nicely see that these core interacts are indeed enriched for proteins that are very likely to be found in such condensates. So what are the biomolecular condensations? I'm talking about this now for a while. So basically, one can envision this as membraneless organelles that are found in the cell in different very vital functions of the cell, including transport, including reaction kinetics, but also in transcription and translation. This condensation is mediated by intrinsically disordered proteins. So those are proteins that have very unstructured domains, such as very well known ones are FUS, WS, or TAP15. So based on this data we acquired so far, we hypothesized that NAP98 fusion proteins specifically alter the nuclear chromosome, and this is leading to the distributional phenotype we know of this type of leukemia. This can be seen like this, that we have this N terminus of NAP98, which is only equivalent glycines, it's the unstructured part, and then we have gene regulated part of the C terminus. And one way to analyze this biomolecular condensation beyond the imaging we've already shown is so-called biotinylated isoxazole, or short BISOX, BISOX, which once treated, once added to a protein lysin, it specifically precipitates those intrinsically disordered proteins and can then further be analyzed in downstream applications such as the Western blot. And here we can nicely see we have found NAP98 kdm5A fusion protein treated with BISOX, and it precipitates in a dose-dependent manner while the control does not precipitate. So this was already another step to answer the question, how is biomolecular condensation involved in this phenotype of leukemia? So what we then went on, we used this assay, this BISOX assay on a human leukemia cell line without our fusion proteins, and submitted it to mass spectrometry and turned it, long name, biotinylated isoxazole mediated condensome mass spectrometry, or short BICON-MS. And by analyzing this cell line without our fusion and just showing how this technique works, we can see that we have again here FUS, TAP15, EWS, and some other proteins that have been described already to be part of biomolecular condensates that are nicely precipitated dose dependently. And if we have a more global view on the data, we can see that in this human leukemia cell line, we find after precipitation with the isox very important members of transcriptional regulation, including the mediator complex, the polymerase II complex, but also coactivators and other very important RNA binding proteins. Setting the technique, we now, of course, perform this on our fusion expressing cells, NAP98 kdm5A and NST1. And here I just have time to give you a very brief and global overview of the data, which should basically show that indeed, so here we have the abundance of the different proteins we find in our masking approach. This is our human leukemia cell line, and here we have the higher concentration of the fusions. And just highlighting those two regions, we see that there are indeed massive changes in the nuclear condensome just by expression of our oncogenic fusion proteins. Moving on, we wanted to investigate now this N-terminal part of the left part of our fusion proteins, this intrinsically disordered part. And for this, we designed artificial fusion proteins. So we basically sat down and said, what do we wish for? And then we made it, which is quite amazing these days. So we have an artificial FG kdm5A fusion protein, which is simply a string of lipid alanine glycines, which does not occur in nature in the patient, but we made it. And then as a control compound, we have an alanine fusion protein. And then we express them also in our cells, and we can see the white is our fusion protein, the alanine is very evenly distributed in the nucleus, while the artificial FG fragment has a very similar appearance as our actual leukemia-causing fusion proteins. So this is a nice observation. What actually happens in the transcription level in gene expression? What does those artificial proteins do to a cell? And that's what we did next. So we used our constructs and we put them in fetal liver cells. So you isolate cells from a mouse, put them in culture, and then you can add different constructs and see how the cell changes. And what we can see, if we have no insertion, just wild type cells, we have a massive change compared to our NAP98 kdm5A fusion, which is expected because this fusion causes leukemia. If we look at the artificial AA kdm5A fusion, it looks very similar to a wild type situation, which shows that this fusion is actually non-functioning most likely. But if we have a look at the FG fusion, which we did, it pretty perfectly resembles the situation of the NAP98 kdm5A fusion, which is here further confirmed by well-described hallmark genes of leukemia, like Cox genes or CDK6. Also on the more global scale, and a pathway analysis, we can see that all the cancer pathways and hematopoietic pathways are equally enriched in the artificial FG and the NAP98 kdm5A fusion setting. So what this shows us is that indeed the FG containing intrinsically disordered regions are able to mimic the endogenous function of the NAP98 part in the context of NAP98 fusion proteins. To sum up now the story, which I was very glad to briefly introduce to you, is that the NAP98 fusion proteins actually preferentially interact with proteins found in biomolecular condensation. They alter the global condensome, just their expression changes the condensome of the cell, and then that this artificial IDRs are indeed able to mimic the leukemia gene signature we see in patients. And this is our model, how we envision it, that in a cell we have our transcription, our DNA, and other proteins that are responsible for the machinery. And if we express our fusion protein, this transcription machinery is altered, it's changed. How this happens is still open, but it's a very exciting field. And this then leads to leukemia. And with this, I would like to thank my lab. I conducted this study at the Vet Vienna with Florian as my supervisor and all the members that participated, and also my current home disease at the Tommaso lab who helped guide me in this study. And you for your attention.
Thank you very much for this excellent work that you have introduced to us. It also landed a landmark publication in National Structural and Molecular Biology and has a great impact on our view on this kind of conversations that we do in cancers. Now we move forward also to a very exciting topic, and the winner and the presenter is Barbara Bachmann, who just launched her presentation. And let me then say a few words to it. Now we move to a slightly different area. I would call it more regenerative medicine. It's about chip technology. And she holds engineering degrees in medical biotechnology from the University of Natural Resources and Life Sciences in Vienna, and in technical chemistry from the Vienna University of Technology. And I think the combination is quite remarkable, and the result is quite remarkable, you see. And during her master's work, she worked with cartilage on a chip. And then during her doctoral studies at the TU, she gained valuable expertise in microfabrication, prototyping, vascular tissue engineering, vascularization on chips, and so on and so forth. And we are very excited to hear more about that. Please, the stage is yours.
So good afternoon, everyone. I will not talk about CAR-T cells. I'm not my PhD thesis was about mechanobiology at microscale, and I used organoleptic technology to apply these mechanobiological views. So just imagine for a moment your body is a very mechanically active system. So for example, your heart is pressing blood through your blood vessels, and by that, all of your cells experience some kind of mechanical actuation. And these mechanical inputs are very characteristic for each tissue and organ type, meaning that your tissue cells react to the mechanical stimulation they get with a specific output. Those can be different kinds of mechanical inputs. For example, substrate stiffness. So your brain is very soft while your bones are very hard, and the cells that live in there recognize this. You can also have dynamic mechanical inputs. So for example, if your blood flows through your vasculature, you will have shear stress on either sides of your blood system. Or for example, if you walk, your cartilage compresses. And your cells actually need all of these different inputs to differentiate and to function physiologically. But if something goes wrong, you can have a pathologic phenotype as well. The problem right now is that current biomedical models do not really represent this mechanical environment. So what you have is monolayer cell cultures and animal models. Both of those have very distinct advantages, but also disadvantages. So for cell cultures, they are human, which is obviously an advantage. But they're often cell lines that are cultivated in this monolayer dishes, meaning they do not feel as if they would be in a body, meaning they also react differently. But those cultures are very fast and cheap, which is why a lot of people use them. On the other side of the spectrum, you have animal models, which are of course not human. But you have a whole organism that has this tissue dynamics and complexity that really represents what cells will be like in a body, but they're usually very slow, pretty expensive. Organ-on-a-chip technology kind of fills the gap between those two by combining all of the advantages. So you can use human parameter cells, and you can arrange them in a way that really represents tissue dynamics and the multicellular architecture. And they're also relatively faster than animal models and relatively cheap because you don't need to use animals, and you can use, for example, plastic components. So, but what is the big advantage of these organ-on-a-chip models? It's that usually in vitro culture, you're really stuck to these plastic tissues. So you have a well plate, and you need to design your experiment according to the well. With organ-on-a-chip technology, you can use microengineering techniques, which means you can actually look at what the cells would experience in the body and design the environment around them. So you can place different cells in different locations, but next to each other, and you can also give them different mechanical inputs. So with that, you can really represent your native tissue niche. Meaning, as I said, you can put multiple different cell types, but you can also cultivate them in the same medium, and you can, for example, produce those chips. And you can also recreate pathologic scenarios. So what I did in my PhD is that I incorporated different mechanical actuations into these organ-on-a-chip models. I actually designed 5 different organ-on-a-chip models that deal with different kinds of mechanical stimulation. For example, I looked into matrix elasticity cartilage-on-a-chip models and also into dynamic mechanical stimulation vasculature-on-a-chip models. So because all of these 5 different models would kind of exceed the 10-minute talk, I'm just going to quickly talk about the vasculature-on-a-chip model. So as you might know, you have 2 different vascular systems in your body. You have the blood vasculature that circulates blood through your blood vessels. And by this circulation, there's actually fluid being pushed out of the blood vessels and into your interstitial tissue. So you have a natural fluid buildup within your tissues. And this is why you have the second vascular system, which is the lymphatic system. And the lymphatic system takes up all of these fluids, and the fluids with the fluids together, also, for example, immune cells, particulates, bacteria, viruses, everything that ends up in your tissue, circulates through the lymphatic system, through lymph nodes, and back into the blood supply. And by this circulation, you have interstitial flow, meaning you have flow towards the lymphatic system, which is what I emulated in my organ-on-a-chip system. So these cells here,
They actually feel if there's a fluid buildup, and if they have a flow towards them, they think, okay, there's too much fluid now, and I need to grow to transport this fluid. So this is what organ-on-chip system can look like. So you have here in red, you have medium reservoirs to feed the cells. And here in the middle, you have these green and blue cell culture chambers, which in my case are filled with blood endothelial cells and lymphatic endothelial cells. And what's the clue about this system is that you have a hydrostatic pressure-driven flow, meaning that here you have a fluid column height of 4 millimeters, whereas here you only have 1 millimeter. And by this fluid height difference, the fluid actually presses itself through these cultures, which results in a directional flow from here to here that is the same magnitude as it would be in the tissue. So this is what it looks like if you look up close. Here again, we have a flow from the blood endothelium to the lymphatic endothelium, as we saw in the schematic before. And we have the flow as it would be in the body from blood to lymphatics. And what I found in my organ-on-chip system is that the cells, they actually react the same way as they would do in the body if you engineer your system like this. So if you cultivate them statically, you don't have any vascular formation. But if you cultivate them dynamically, they actually form vasculature, so they respond. If you give them an environment, it's the same as in the body, they respond the same way as they would in the body. So with that, I'm already at the end. To sum up, I engineered 5 different kinds of those systems and actually looked into different mechanical stimulations. And with those organ-on-chip systems I engineered in my thesis, we can actually use them for cartilage and regeneration studies. So, for example, if you hurt your knee skiing, we can look into what would make your cartilage regenerate. We can also look into how to more efficiently vascularize tissue engineered constructs. So if you want to grow liver, how can we actually make a vascularized liver that can grow to the size as you need it to be? And with the last system, we can also look into lymphatic targeted drug delivery, meaning that you can get, for example, a vaccine exactly into the lymphatic system where you would recognize the virus. And I would like to thank the supervisor for the team, and also our collaborators at Trauma. And there won't be any questions, which is nice.
Thank you very much for this beautiful example of our thesis, PhD thesis in applied research, fully meeting the criteria of this category for prizes. Congratulations once again. Now, we're coming somehow to an end here of this ceremony. And let me just give you a brief outlook on what is ahead of us with this online meeting. Now, the 13th annual meeting has 15 scientific sessions that are distributed over these afternoons that are shown here in this table. What is important for you to know is registration is possible anytime. You can join in anytime, and there are no fees for members and very low fees for non-members. So please join. It's an extraordinary program this year. And with that, I would like to thank all of you for a most entertaining, wonderful scientific afternoon. Thank you again to our sponsors, to Dr. Unterer, to Chantal, and Professor Kattinger, for coming, for sharing your valuable time, and for your sympathy that you support us for so many years. I invite our on-site guests for a little come together according to all regulations and 3G rules with distance measures and so on. And from you online on YouTube and Zoom, I would like to say bye now. Thank you for joining in, and we are looking forward to seeing you again in our online sessions. Thank you.